The next set of amino acids that we're going to focus on will be the branched chain amino acids leucine, isoleucine, and valine. Now, the first thing you have to know about these amino acids is the following. Not only can our hepatocytes, our liver cells, metabolize these amino acids for energy, but our muscle cells can also actually break down these same amino acids. Now, the focus of this lecture will be the metabolism of leucine, but we'll also briefly talk about valine and isoleucine. And at the end, we're going to talk about a disease in humans that affects this particular pathway, or actually the pathway involved in metabolism of branched chain amino acids, known as maple syrup urine disease. So let's begin with step one. So in step one, we basically want to remove that alpha amino group. So this reaction is known as a transamination reaction. So we basically begin with leucine, and we have this alpha amino group. So we have an enzyme, an aminotransferase known as branched chain aminotransferase, catalyzes the movement of this alpha amino group from the leucine onto alpha ketoglutarate, and we form glutamate. Now we also attach an oxygen onto this carbon to form this alpha keto isocapriot intermediate. And just like any other aminotransferase, this one also utilizes PLP, pyridoxal phosphate. Now, once we form this alpha keto acid intermediate, the enzyme that acts on this, the next enzyme that acts on this, is an enzyme found in the inner mitochondrial membrane, an enzyme known as branched chain alpha keto acid dehydrogenase complex. Now, this complex is homologous to two other complexes that we discussed before. So we talked about pyruvate dehydrogenase complex, and we also talked about alpha ketoglutarate dehydrogenase complex. And just like those two complexes, this one is homologous. So it consists of 3 catalytic subunits, and it utilizes 5 cofactors. So we have thiamine pyrophosphate, NAD+, FAD, we have lipoate, and we have coenzyme A. And so what this enzyme does is it basically oxidatively decarboxylates this molecule. So it takes off this carbon dioxide and replaces it with a coenzyme A molecule. So we form a coenzyme A intermediate known as isovaleryl coenzyme A. Now, once we form this, next we have a dehydrogenation step. And the enzyme that catalyzes this is isovaleryl coenzyme A dehydrogenase. So what it does is it uses an FAD to basically abstract an H atom from this carbon and an H atom from this carbon. So it essentially oxidates this molecule. It reduces the FAD into FADH2, and the 2 electrons here that are left over are used to form a pi bond between this carbon and this carbon. And so we transform this into beta-methylcrotonil coenzyme A. So we have a transamination step, an oxidative decarboxylation step, we have a dehydrogenation step, and now we're going to look at the 4th step, a carboxylation step. Now, any carboxylation reaction basically utilizes an enzyme known as a carboxylase. And a carboxylase needs 3 things. A biotin molecule, it needs a carbon source, carbon dioxide, and it needs an energy source, ATP. So the enzyme beta-methylcrotonil coenzyme A carboxylase contains biotin as a prosthetic group that aids this reaction. And it basically hydrolyzes ATP, and the energy released in the hydrolysis of ATP into ADP and PI basically allows us to attach a carbon dioxide onto this carbon here. And we form the beta-methylglucoconil coenzyme A molecule. Now, once we form this, the next step is to basically lyse. So we want to break this pi bond, and then we want to attach a hydroxide group onto this carbon and an H atom onto this carbon here. So we basically have an incoming water molecule is used by an enzyme called methylglucoconil coenzyme A hydratase to basically form a group, the hydroxyl group here in this orientation, and form an H atom in this orientation as shown here. So this molecule we actually saw before when we discussed ketone body synthesis. So we have 3-hydroxy, 3-methyl, glutaryl coenzyme A. And once we form this molecule, the final step, step 6, is a lysing step. So we basically want to lyse, break this bond here, and form these two final products. So we use an enzyme, HMG coenzyme A lyase, where HMG stands for hydroxymethylglutaril. And this enzyme breaks this bond here. It forms acetoacetate and acetyl coenzyme A. So this is our 6-step process. So again, a transamination, then we have an oxidative decarboxylation, followed by dehydrogenation, forming of this pi bond. Then we have a carboxylation, attaching of a carbon dioxide. Then we have this simple hydration step, where we break the pi bond, attach a hydride. And finally, we have a lysing step. We basically cleave the sigma bond, releasing, forming these two molecules. And now these two molecules can basically be used to generate energy by the cell. And this happens not only in our liver cells, but also in our muscle cells. Now, what about metabolism of valine and isoleucine? Well, the metabolism of these two branching amino acids is very similar to the metabolism of leucine. So what do we mean? Well, basically, in the first step, in either one of these two amino acids, we also have a transamination step to form an alpha keto acid intermediate. Once we form the alpha keto acid intermediate, this same dehydrogenase complex also acts on these alpha keto acid intermediates to form a coenzyme A derivative. And once we form coenzyme A derivatives, we follow a similar set of steps to basically form these similar molecules, except in the case of valine, we form propionyl coenzyme A and carbon dioxide. In the case of isoleucine, we form acetyl coenzyme A and propionyl coenzyme A, as shown on the board. So by metabolizing leucine, we form acetyl coenzyme A and acetoacetate. By metabolizing valine, we form propionyl coenzyme A and carbon dioxide. By metabolizing isoleucine, we form propionyl coenzyme A and acetyl coenzyme A. Now, let's take a look at a disease that exists in humans known as maple syrup urine disease. And this disease is basically a disease of this step here. So, if a person has this disease, the branched-chain amino acid can basically undergo step 1, but it cannot undergo step 2. And that's because there's either an absence or some type of deficiency of this enzyme here. So this is an autosomal recessive disease. And what that means is, for a person to have this disease, they have to inherit 2 defective traits, so 2 defective genes. So one defective gene comes from the parent, the male parent, and one comes from the female parent. So if this enzyme doesn't function correctly, then what happens is we're going to see a buildup of not only branched-chain amino acids, but also alpha keto acids, these intermediate molecules here. So we're going to accumulate them, build them in our, we're going to see a buildup in the blood, in our urine, and ultimately this disease is called maple syrup urine disease because the urine of a patient with this disease smells like maple syrup as a result of that high level of the alpha keto acids. So maple syrup urine disease is an autosomal recessive condition in which there is an absent or defective branched-chain alpha keto acid dehydrogenase complex. This causes a buildup and elevation of the alpha keto acids as well as of the branched-chain amino acids. And this ultimately leads to not only mental problems, but also physical problems. Now, how can we diagnose a patient with this particular problem? Well, basically, we can take a urine sample and mix the urine sample with a molecule known as 2,4-dinitrophenyl hydrazine, which basically looks like this. So why? Well, we know there will be a buildup of these alpha keto acids within the urine of that patient. And by mixing the urine with this molecule, we're going to see this reaction take place. This will basically attach onto this carbon, replacing this oxygen. So we'll form the 2,4-dinitrophenylhydrazone product. And so this molecule will ultimately precipitate within the urine, and that can be used as a diagnosing factor. So to summarize, we see that we have 3 branched chain amino acids: leucine, isoleucine, and valine, and these can be metabolized, as discussed in this lecture, not only in our liver cells, but also in our muscle cells. Now, if we have a defect in this complex, this is known as maple syrup urine disease, and this causes an elevation of not only these alpha keto acids, but also an elevation of the branched chain amino acids in our urine and in our blood.